Welcome to ToToZ Collection Biochemistry session, and in this session we're going to zero in on carbohydrates. Now these are the same carbohydrates that you dealt with at non-quota, but we're just going to look at them from a medical point of view. And for you who didn't do non-quota, just follow me really well. Write down something as you're watching this video and you have carbohydrates at the tips of your finger. So we're going to start with the function of carbohydrates, and the first one that we've been told from grade 1 was carbohydrates are energy-giving food. But here we're going to delve a little deeper into exactly how much energy do we get from carbohydrates. So from carbohydrates we get about 4 kilocalorie. We get about 4 kilo. All right. Okay. Yeah, so we get about 4 kilocalorie per gram. Okay, 4 kilocalorie per gram. So the units are important. So kilocalorie per gram. So that is specifically from carbohydrates. But in times of starvation, your body is going to resource to other means of energy if your carbohydrates are insufficient in your body. So next up, if carbohydrates are completely depleted from your body is going to go to your fats. And then from your fats you get about 9 kilocalorie per gram. 9 kilocalorie per gram. And then if your fats are depleted, then your body will finally go to proteins, and from your proteins basically produce the same amount of energy as carbohydrates. So that is 4 kilocalorie per gram. 4 kilocalorie per gram. All right, so we have 4 here, we have 4 here, and then we have 9 here. But we would ask ourselves to say, okay, fats are producing the highest amount of energy, but why does the body still start with carbohydrate initially? Okay, so this is pretty simple. So this is because utilization. To produce our energy, we must always use oxygen. Okay, in spite of whatever means of energy production, whether we're using proteins, whether we're using fat, or whether we're using carbohydrates, we must use oxygen at a certain point in all the processes. So carbohydrates use the least amount of oxygen. Okay, carbohydrates use the least amount of oxygen, so they are more like so I don't know how you would say it in English, but yeah, in simple English, carbohydrates use the least amount of energy, and that is why they're preferred first as the main source of energy compared to these others. Okay, so that's one. And then the other thing that we can say is that there is a common term that says fats burn in a carbohydrate flame. All right, so they say your fats burn in a carbohydrate flame. Carbohydrate flame. So this means that to completely break down your fat to give you your energy, at one point you must use a carbohydrate. Okay, so it's more like the fat break it down to make your energy is highly dependent on your carbohydrates. So there is an intermediate called oxaloacetate that we need, and this oxaloacetate is gotten from our carbohydrates, and this oxaloacetate is important in the fat breakdown to produce our energy. So even if we were to lean to the fat metabolism for us to produce energy, at a certain point we'll still need the carbohydrate. Yeah, so we see that the carbohydrates are very much important. So why waste that energy of using two things at once to produce our energy? Why not just stick to one thing if that one thing is available? So that's why the body very much likes carbohydrates as a production of energy. Yeah, so we have that being said, and at this point we can already answer tons of questions in our exam paper. Okay, so we continue on by saying carbohydrates, they can combine with proteins. Okay, so you have a carbohydrate, it makes a conjugate with a protein, and now it is called a glycoprotein. It's going to be called a glycoprotein. If that carbohydrate combines with a lipid, it's going to be called a glycolipid. Okay, and these glycoproteins, glycolipids, you find them on the cell membrane, performing different roles. So some of them can be receptors, and some of them can be antibodies or antigens or anything. They can serve multiple roles on your cell membrane. Yeah, and then we also get to see that excess carbohydrate is converted to fat. Yeah, excess carbohydrates is converted to fat. And then aside from that, we also see that carbohydrates form a structural basis of many organisms. So we see the carbohydrates forming the cell wall of plant cells. So that is what? Cellulose, eh? Cellulose forms the cell wall. And then we also have another one called chitin. You guys who went to private schools would call it chitin, however you pronounce it. Yeah, but this one forms what? The exoskeleton of insects. So that is what we're saying, the structural basis. So these are more like the high-yield functions of carbohydrates. Of course, you could add more, but these are the main ones that I wanted you to focus on, and for you to be able to have examples on them, and just for you to be able to play around with them. All right, just in case I would ask you any questions on them. Yeah, so we have that. So the next thing that we can look at is a general formula for carbohydrates. Now, these carbohydrates come in different classifications. That is one thing we should note. So we have the carbohydrates. That have one sugar unit, one sugar molecule, and we call those monosaccharides. We have the carbohydrates that have two sugar units, we call them disaccharides. And then if your carbohydrate is having sugar units ranging from 3 to 10, then that is going to be an oligosaccharide. And then if it is having from 10 beyond, this is going to be a polysaccharide. So monosaccharide was saying there's just one sugar unit, but these guys were saying 2 and beyond. So these 2 and beyond are going to be joined together via some linkages, via a glycosidic linkage or a glycosidic linkage. So with your monosaccharide, this formula that you're seeing here only applies with monosaccharides. Okay, because basically, how do you we're saying carbohydrate, and if you were to break this word down, this would just be a hydrated carbon. Hydrated carbon. So the hydrated part is the H2O part, and then the carbon part is that carbon part outside. So the general formula is going to be split in that same rationale. So you're going to have our CN and our H2ON. Okay, so if we put 2 here, general formula. This is how we play. So let's think of a carbohydrate. Let's say we have glucose. So glucose is a 6 carbon carbohydrate. So meaning I'm going to put 6 here and I'm going to put 6 here. So this is going to be C6 and then H12 and then O6. This is basically what this means. Okay, aside that, I can also think of saying, all right, what if I were to say maybe pentose? That's 5. So this is going to be C5 here, and this is going to be H10 there, and then O5 there. So this is how I write my chemical formula. But yeah. So this general formula only applies with monosaccharides. What applies with disaccharides? Check out the next video. So this one is strictly on monosaccharides. Sorry, I forgot to mention that this video is strictly on monosaccharides. For disaccharides and the oligo and the polysaccharides, check out my next video, probably next week. Okay, so here we're going to zero in on this. Okay, and there we have different examples where we have our glucose, fructose, galactose, and we're going to see how these are different from each other. Yeah. And then here we have our sucrose, lactose, maltose, raffinose, stachyose, our starch, our glycogen, our cellulose. They're all going to be there. Okay, and here we're appreciating the monosaccharides based on the number of carbon atoms, and they actually have names. Yeah, they actually have names. So if your carbohydrate has got 3 carbon atoms, we're going to call that a triose. We skip 4. We're not interested in 4. There are not much cool examples under 4. We go to 5. And if it has 5 carbon atoms in its structure, we're going to call it a pentose. If it has 6, we're going to call it a hexose. If it has 7, we're going to call it a heptose. It just keeps on going. It just keeps on going. All right. So yes. So 3 trios, 5 pentose, 6 hexose, and we must have examples under each. So under 3 we have glyceraldehyde, and then we also have dihydroxyacetone. Know the examples. So 3 we have glyceraldehyde and dihydroxyacetone. So where is this glyceraldehyde coming from? Where is this dihydroxyacetone coming from? So this is coming from a process called glycolysis, which is basically the breakdown of your glucose. Glycolysis. So you're going to know this process in your third year, but for now just know that here you are changing your glucose into a compound called pyruvate. Just know this. And in the absence of oxygen, which is anaerobic respiration, you're not going to have pyruvate as a final molecule. You're going to have lactate or lactic acid as a final molecule. Yeah, lactic acid. Yeah, this is glycolysis. So among the intermediates of this process that we're calling glycolysis, there is a guy called fructose 1,6-bisphosphate. So this fructose 1,6-bisphosphate is split by an enzyme called aldolase. So this one here, sorry. So we're saying fructose 1,6-bisphosphate being broken down by the enzyme aldolase, and then it gives you two things. So it gives you your glyceraldehyde, and it also gives you your dihydroxyacetone. Okay, so what does this mean? So this means that this fructose 1,6-bisphosphate is actually a 6-carbon molecule, and then once aldolase breaks down this guy, it changes into a 3-carbon molecule and a 3-carbon molecule. And what are we calling these 3-carbon molecules? We're calling them trios and trios. Yeah, so it's basically what I need you to know, and that's why it's saying an initial breakdown of glucose product, an initial breakdown product of glucose. Yeah, because it's along the pathway of glycolysis where we break down glucose into pyruvate or lactic acid. And then note this structural difference here. We're going to talk about it later. Yeah, and from there we go further by saying, all right, we also have pentose, and in this pentose we are going to have a guy called ribose, and ribose is a component of RNA and DNA, and we're going to delve deeply into that in nucleic acids. But for now, just know that, okay, we have ribose, and ribose is a pentose sugar, which is a 5-carbon sugar, and yeah, so its counterpart is going to be ribulose, which is an intermediate of photosynthesis. And again, note the structural difference here. High-yield, we're going to talk about it later. We're going further by looking at glucose, and we see how glucose is similar to galactose, and how we also have fructose. So I think these are familiar. So again, note the structural differences. We're going to talk about them later. All right, so that's just what I wanted you to know. And then here we have the isomers of carbohydrates. So did I mention that carbohydrates is a work in the pack? So did I add that? Okay, this pack hasn't been cleaned, so there are a bit of like stones in the pack, there are a bit of like bottles in the pack, so you might prick yourself, all right? So you might hurt your toes if you're not careful. So these are the toes, these that you're seeing here, isomers, Ishman. If you don't understand this, trust me, you're going to be lost. Focus. Get your pen, write it down, draw diagrams, concept diagrams, and yeah, just lock in. So the first thing that we're going to talk about are let's start with our functional isomers. So with our functional isomers, we have a group called a carbonyl group. So a carbonyl group is just where you have your carbon double bonded to an oxygen. We're going to call that a carbonyl group. Okay, carbonyl group. Now this carbonyl group can be at the end of a molecular structure, and then the same carbonyl group can be within the length of your chemical structure. Okay, so two scenarios. So a scenario where it's at the end, and then a scenario where it is within the length. So if it is at the end, we're going to call it a aldehyde. If it is within the length, we're going to call it a ketone. We're going to call it a ketone. So you've seen that this R here, this R means rest of the chain, even this R means rest of the chain. So meaning this carbonyl group is within the length of the molecule. But here we only have the rest of the chain being on one side, meaning that this carbonyl group is at the end of a carbonyl chain. Okay, and then the other mnemonic that you can use to Swatila this is that here you have this word ketone. So ketone, we're saying if it is within. So someone did tell me that imagine your carbonyl group doesn't want to be alone. So it doesn't want to be alone, so it sticks itself within the length of the chain. So here you're going to have a carbonyl group. So chain here, carbonyl group at the center. It doesn't want to be alone, so that's why we call this a ketone, because it doesn't want to be alone. But then the aldehyde group is chilled. It can stay at the end of the chain. It doesn't kill. And then when you look at specific examples, so here we're looking at carbohydrates. So we're going to look at carbohydrate molecules that can either be either have a aldehyde group or a ketone group. So here we're saying that the aldehyde group exists at the end of the chain, and then here we're seeing that the end of the chain is right about here, but we can have the carbonyl group being within the length of the chain, and here we see it being in the middle here, so we're going to call it a ketone group. Okay, so this is what we call an aldose, and this is what you call a ketose. Now remember that here we're talking about isomers, and with isomers we're talking of how the molecules are going to be different in terms of their structure. So glucose and fructose are both hexose carbohydrates. Hexose meaning they have 6 carbon molecules. But what is the difference? The difference is that one has an aldehyde group while the other has a ketone group. So one is an aldose and then the other is a ketose. Okay, so which one is the aldose? It's glucose. Which one is the ketose? That's fructose. Very, very high yield. Please keep this in mind. And we're going to talk about what this D and what this D means later, but for now just keep in mind that glucose and fructose are cousins. How? One has a aldehyde group, the other has a ketone group. What is an aldehyde group? What is a ketone group? That is all up to you. I think I have explained that really, really well. Okay, yeah. All right, so that was your functional isomers. So we can take that guy there. Functional isomers, done. From there, we can go to epimers. Epimers, which are also called diastereoisomers. Epimers. Okay, so what are epimers? So epimers, these are just molecular structures that differ at one point, at one carbon atom, anywhere within the carbon atom. They just differ. Okay, so chemical formula is going to be the same. The chemical formula, the CH shani, so it's going to be the same. C6, H12, O6, it's going to be the same. But when you draw them linearly, they are going to be something else. They're going to be different from each other. Okay, so this is where we have the examples mannose, glucose, and galactose. Now we're going to talk about the D later. For now, let's just focus on the mannose, glucose, and galactose. All right, so these guys are epimers of each other, or they are diastereoisomers. So what does this mean again? All right, so these carbon atoms that you're seeing here, they're named or they're numbered. So here we see the numbers starting from 1 there, 2 there, 3 there, 4 there, 5 there, and 6 there. They're numbered in that order. Okay, and then keep in mind that glucose is going to be our point of reference. Glucose will always be our point of reference. It is the supreme hexose. Also, these guys are also hexose. This guy is also a hexose, just as fructose was a hexose. So with glucose, we're saying that this is the original structure of glucose. Original structure of glucose. So pay close attention to the hydroxyl groups. Carbon number 1 doesn't have a hydroxyl group. Carbon number 1 in glucose has a aldehyde group. A hydroxide group looks like this, not like this. Keep that in mind. So here we have OH being on the right side, OH being on the left side, OH being on the right side, right side. Okay, so it goes right, left, right, right. Okay, and then this one here, right? Yeah, we can include it there. Okay, so now compare that to mannose. Mannose, what do you see? What is the difference? The difference is that the first OH is on the left. There is a difference. So just by that difference, this guy qualifies to be epimers of each other. Epimers of each other. Why? Because they differ at a certain carbon atom. Their structure differs at a certain carbon atom, but their chemical formula remains the same. So in spite of you changing the OH from this side to this side, mannose and glucose will still be 6C6H12O6. Okay, so here we can say that glucose and mannose differ at carbon 2. Okay, now compare glucose with galactose. What is the difference? All right, so this is going to be at carbon what? 4. Okay, going to be a carbon 4. So in summary, glucose being the original hexose is an epimer with mannose at C2, and then it is also an epimer with galactose at C4. Yeah, so we have that. Oh, and by the way, you should be able to draw glucose from your head. That is a very high-yield examinable question where they tell you to draw the structure of glucose from your head. So you're supposed to know this linear structure. And then once you know that linear structure, just know that if you change the orientation of the OH, take it to the left side at C2, you've made mannose. Bring back that OH to the right side, but change the OH from on C4, then you're going to have galactose. Right. So again, more examples of epimers. So there we're looking at glucose. So here we can look at a different guy. Let's look at a pentose. This is a pentose, eh? How many carbons? 1, 2, 3, 4, 5. Yes, pentose. So the pentose here that we have is our ribose, but then we can also see that ribose can also have epimers such as arabinose, xylose, and lysose. And you can look at the structural differences that we're seeing here. So with ribose, all the OHs are on the right side, okay? But with arabinose, the first OH is on the left side. So this is at C2, okay? With xylose at C3, we're going to have our OH being on the left side. With xylose, you have both C2 and C3 being on the left side. Epimers. Yeah. And from there, from our epimers, so we've looked at functional isomers, we've looked at epimers. What haven't we looked at? Enantiomers and anomers. All right, but before we look at that, let's just appreciate this thing called the ring formation. So from the previous structures that we're seeing, we're looking at the carbohydrates as a linear structure, but we can also have these carbohydrates existing as a cyclic structure. And how does it happen? So with carbohydrates, mostly the 6 carbon molecule carbohydrates, you're going to see the ring structure forming. Now this ring structure forming takes place between carbon 1 and carbon 5. Carbon 1 and the carbon 5. Carbon 1, carbon 5. Carbon 1 and carbon 5. Yeah. So with carbon 1 and carbon 5, we're going to have them attaching, like from there to there, they attach. And then you see this guy here. This guy here is going to remain on top there. Ooh. This guy here remains at the top there. Okay, so that's from carbon 1 to carbon 5. So we do have these two words here called hemiacetal and hemiketo. And remember here we have glucose and fructose. What's the difference between glucose and fructose? One is the aldose, one is the ketose. Yeah, so here with glucose is a aldehyde. So when we have a aldehyde from glucose mixing with this alcohol group here, so the OH is the alcohol group. We call that a hemiacetal formation or a hemiacetal linkage. Keep that in mind. Write it down, hemiacetal. And then we can also have our ketone. Our ketone forming a bond with the hydroxyl group here. So this one is going to be from what? C2 to C5. C2 to C5. So if we have our ketone bonding or making a bond with our alcohol at C5, we're going to call that a hemiketo formation or hemiketo linkage. So keep that in mind. So we're going to make this guy here and we're going to make this guy here. So upon us forming this cyclic structure, this is where we have a term called anomers. So there are two types of anomers. So we have the alpha and then we have the beta anomers. So these anomers work like this, huh? These anomers work like this. So have we seen this carbon here? So if you have this carbon that's attached to the OH, that's double bonded to the OH. So the carbon that forms your carbonyl group, it has a name. The carbon that forms the carbonyl group has a name. So we call this the anomeric carbon. The anomeric carbon, the carbon that has that forms your carbonyl group, we call it the anomeric carbon. Yeah, so here, which one is our anomeric carbon? C1 in glucose, and here it is C2 in fructose. Okay, this is where we have the concept of the anomers coming in. Anomers. So when you see that on your anomeric carbon, once you form your cyclic structure, the OH is pointing downwards. We call that an alpha carbohydrate. So even this one is a alpha carbohydrate. Okay, so we can look at it from this point of view. So we have the same glucose. So we have the same glucose here. But then, so we have the same glucose here. So this glucose forming a cyclic structure, and the OH is pointing downwards. At your anomeric carbon, C1 in glucose, we're going to call that a alpha glucose. And then here your OH is pointing upwards, so we're going to call that a beta glucose. Yeah, so alpha, beta. So alpha pointing downwards, beta pointing upwards. So you have that. So these are what you call anomers. So what are anomers? Anomers are molecules that differ at the anomeric carbon, that differ in structure at the anomeric carbon. Who is your anomeric carbon? This is the carbon atom that forms your carbonyl group, or that makes up your carbonyl group, or that participates as your carbonyl group. Yeah, something like that. And lastly, we can talk of these guys called enantiomers. I think I forgot to add an image about enantiomers. We can talk about them regardless. Enantiomers. So when we talk of enantiomers, we're talking of this is where we have our D series and our L series. So remember that D glucose, the one that I told you we'll talk about later. We're going to talk about it right now. So D. Okay. So check this is how she goes. So we have D and then we have L. So first things first is that you're going to swatila that L is for left. Let's start from there. L is for left. Let's lay that and then talk about it a little bit further later on. Yeah, so D and L, so D being for left. So with enantiomers follow a concept called optical isomerism of each other. So this is where you have what you call polarized light being projected to your glucose or to your glucose solution, and then this glucose can either deflect this polarized light to the left, or it can deflect the polarized light to the right. Okay, so if it does do that to the left, you call that a L glucose. If it does do that to the right, you're going to call that a D glucose. Now it goes beyond optic isomerism and just comes down to the structure. So structure wise, you can talk of it in this manner. So this is the best example here. So here we have our D glucose, then here we have our L glucose. O club. Yeah. So here we have our D glucose, and then here we have our L glucose. So the D glucose, we are saying that it is to the right. Our L glucose, we are saying it is to the left. Now, what is to the left? What is to the right? Structure-wise, here is that we have this OH, this OH, this OH. Now, which OH are we going to follow to see that, okay, this OH is on the left side, this OH is on the right side? So we are going to follow the OH that is furthest from the aldehyde group, but not the last one. That is the exception. The OH furthest from the aldehyde group, but except the last one, not the last one. Okay, so just say the second last OH, or sorry, the second last carbon atom from your aldehyde group. So the second last carbon, which is going to be C5 in this case. Now look at its orientation of the OH, of the hydroxide. Is it on the right side? If it is on the right side, you call that a D glucose. If it is on the left side, you call that a L glucose. So D glucose, L glucose, these are enantiomers of each other. And what property are they following? They are following the property of optic isomerism. So you can call them enantiomers of each other. You can also call them optic isomers. Optic isomers of each other. So this is your D and this is your L. Right. Once you know that, this sums up our lesson for today. So let's hope you have appreciated the enantiomers with examples. Let's hope you've appreciated the epimers and diester isomers with examples, functional isomers and anomers. Yeah, so this is specifically under the topic of isomers of carbohydrates. So this was just the basic intro, monosaccharides. I haven't even talked about any related conditions to each of these carbohydrates. I think I'll do that in the notes, yeah, to save up our video time, video space. Yeah, the notes should have them.